opportunity to be here today. I'm excited to talk to you a little bit more about how we explore caves beyond Earth. I know Diana, who's really amazing cave scientist, gave a talk a few months ago. And so I'm trying very hard not to overlap on some of the things I think she talked about, but also provide enough background that if you missed that particular talk, we don't get you lost in the lurch. So if you have questions along the way, of course, please let me know and I'll try to fill in any blanks I don't cover. Okay, so how do we look for life on Mars? It turns out that, let me get my slide to move forward, that Mars's surface used to have, a pretty dynamic hydrologic system in the very earliest part of its history. So we're talking 4.1 to 3.7 billion years ago. And so you have these, rivers and lakes and potentially even an ocean, depending on who you talk to, and a beautiful groundwater system that's interacting with that surface system. As you move forward in Mars's history, as the magnetic dynamo begins to fail and we start losing atmosphere, it starts getting colder and drier. And so more and more of that water gets locked up either at the surface in ice or in the subsurface as ice, with much less of the water in restricted lakes and stream systems and in the subsurface. And as you move up towards the end of that first billion years of Mars's history, you're pretty cold and dry at this point. If there is liquid water at the surface, it's really restricted to very concentrated brines that are intermittently stable. And any water that you have that's liquid is primarily in the subsurface. And those conditions remain the conditions that we're operating under on Mars today. So in the first billion years of Earth's history was when we kicked off life, right? In the first billion years of Mars's history, where we start having a divergent trajectory in terms of water compared to Earth. And so because of that, any life that we might want to look for we're best off looking in the subsurface. So if the majority of Mars's water is now in the subsurface, outside of the poles at least, how do we get there? And the answer is that there are a couple of options. You can drill, which has its own set of challenges, which we've seen the Phillips-Lander face. You can also take advantage of natural subsurface access points. So these are all the black dots that you see on this map of Mars are subsurface access points that have been identified in what's called the Mars Global Cave Candidate Catalog that Glenn Cushing put together and that has been built on since then. These access points may be caves. They may also be pit crater chains, so just vertical collapse features. So what does that look like? Well, pit craters can form in a number of ways, all of which really are related to loss of support for surface material and collapse. And in the example I'm showing on the left-hand side, you can see a normal fault. So the crust has pulled apart here on Mars. And because of that, you have a series of collapse features in a line related to that fault. And so in the middle, you have a block diagram of what that process looks like to kind of give you a three-dimensional perspective on how some of these collapse features might form. There are other ways collapse features might form, including dike intrusions, where, you know, once the magma is removed from the system, it can no longer support the ground above it, and so you get a collapse. You can also have constructional features. So on the left-hand side, all collapse features. On the right-hand side, we have constructional features. And so this is a beautiful image. It's one of my all-time favorite Mars images, showing the lava tubes running across the surface of Mars. This was taken by Mars Express. And so how do these constructional features form? So initially, when you have a volcanic eruption, and in this case, we're talking about a low viscosity volcanic eruption, right? Because we're getting a flow instead of an explosion. That flow starts running across the surface, and over time, it starts getting more and more channelized. And the lava itself is very hot. You know, when it's coming out of the volcano, it's close to 1200 degrees C, but it's in contact with very cold atmosphere. And so because of that, over time, you start to form a crust over that lava. So essentially the top part of that surface solidifies. And you can actually see in the two images on the right-hand side of this frame, which are both taken in Hawaii, what that looks like. So you have the crust that those guys are very precariously standing on over the active flow that is flowing beneath them. I don't necessarily recommend doing this, but hey, science, right? And so eventually that lava will drain away, right? The eruption ends, and so it's no longer sustained, so it drains away. And when it drains away, you are left with a void space. And so on the bottom here, we have a couple of pictures. The left-hand side is my field assistant, Kayla Miller, and I screaming outside of a cave called Screaming Jaws of Death at Craters of the Moon National Monument in Idaho. And so that's what the outside entrance looks like. And the inside is the image sort of in the middle on the bottom. And you can actually see the cave sort of falling away towards where I'm sitting taking the picture inside the cave. And that reflection in the middle is not actually liquid water, it's just straight up a reflection of the surface conditions. But so that's how lava caves form, or what we call lava caves on Mars. So we see these features on Earth, and we also see them on Mars. And so if we look at the Tharsis region, there's been some great work done both with MARS's, which is a subsurface radar on Mars Express, and SHARAD, which is a radar system on Mars Reconnaissance Orbiter. Both those instruments were designed to actually look for water in the subsurface on Mars, but they've also been used to understand more about these lava tubes. And so if we look at Alba Mons, which is this enormous shield volcano, you can actually see the lava tube tracks. That's what the image on the right-hand side shows, along with their entrances. And when they actually mapped these with the radar system, they found that these tubes may have void spaces up to 8 meters in diameter, so the chamber size, which is enormous. And they may be as shallow as within 12 meters of the surface for really long distances, because of course these flows are flowing at very low angles. So we have laterally continuous tubes that have flown over long distances that are enormous. And that's a really good thing, because it means that there'd be plenty of void space available to explore and to collect water and potentially be a habitat for life, both, you know, in Mars's history and potentially in the future with human habitation. So do these caves actually have water? we'll focus again on that Tharsis region. I just want to kind of keep you oriented in space. So that's that black box on the left-hand side of the screen. And you can see some work done by Casanelli and Head in 2019 that mapped out potential glaciation that persisted through a good chunk of the Amazonian period, which is the period we're in now on Mars's history, although these features are not currently glaciated. And so they proposed that there was an active glaciovolcanic system. So something akin to Iceland, where you have ice coverage, but you also have the heat available from the magma at depth, and you have occasional eruptions that essentially melt the ice. So if you look at the bottom left-hand picture, we start melting the bottom part of the ice and forming kind of a low temperature hydrothermal system associated with that initial little bit of eruption. As that eruption continues, eventually you melt a significantly large area, and you can create a hydrothermal system in the long run associated with those volcanic edifices. So the majority of the surface could still be covered with ice, but you could still have these hydrothermal systems and groundwater circulation associated with those hydrothermal systems. And so if you look at the top picture here, we have an image of the Krafla geothermal plant. And so the surface, as you can tell, is covered with, snow. It's wintertime, but you have this dynamic hydrothermal system in the subsurface, and the energy available from the heat of that magma in the subsurface that is actually providing energy to Iceland. So it's actually quite a lot of energy, and thus great potential to support life. In addition to that, work that Kai Williams did mapped ice accumulations on Mars, and he argues that you could have significant ice accumulating in the subsurface of these lava tube caves, and that they essentially act as cold traps, which would trap water from the atmosphere. I think someone's unmuted. Could trap water from the atmosphere and actually accumulate additional water resources in the form of ice in the caves. And you can see the significant number of these cave entrances are in places where we could be accumulating ice even today. So there might actually be quite a bit of water associated with Martian caves. So we've established that there's water, and NASA says we should follow the water to look for life, because life, as we understand it, lives by diffusion. And so that requires water as a medium for life as we know it. So if we look for life in the subsurface, what exactly do we look for? So my friend Mark Nouveau made this amazing paper a few years ago, where he made a ladder of life detection as a guidepost for future missions. And so you can see the life bar gets from very green, so very strongly indicator of life, to almost white at the bottom. And then you get into what's called suspicious biomaterials below that. So these are things where we have seen evidence on Earth of like a biofabric or metal cycling by microorganisms, or compounds that might be indicative of life, but might also be found in a meteorite, right? And then as you move up the ladder of life detection, you get into molecular complexity and chirality, which is what the little hands mean. Biomolecules like DNA and RNA that provide structure and function, and then into metabolic activity, and ultimately observing growth and reproduction and evolution through time. But a lot of these things are challenging, particularly once you get into the suspicious biomaterials category, because they could be life. It's possible. They could also be an abiotic process because life exploits the same chemical processes that occur abiotically. Life just tends to do them faster. And so it is actually incredibly challenging to look for life. Now, when we've looked on Earth, a lot of the work that has been done in lava tubes focuses on microbial mats and speleothems. So these are visible indicators of life. So you could walk through a cave, see something like this gorgeous snotite here, which gorgeous snotite sounds really odd. Do you know what I mean? And go, oh, yes, definitely life, right? But one of the challenges is that these types of features are randomly distributed in caves, and as far as we can tell, and they don't form everywhere. So what if in a low biomass environment, they don't form? What if, for whatever reason, they don't form on Mars at all? What do we do if we can't see the life that we're looking for? And that may seem like an academic question, but it really gets at a fundamental question that we encountered when we were thinking about MACIE, or the Mars Astrobiology Cave and Internal Habitability Explorer, which is the mission concept that my team worked on for the decadal survey. And none of us could really answer the question for the engineers of exactly how far do you walk in a Mars cave to capture evidence of life, because we were all really focused on, well, we don't know where the mats are going to be. And it was interesting because I had this study going on at the very same time, and in the moment didn't put it together. So maybe that's a failing on me. But if you look at the picture on the right-hand side, this doesn't look like a pretty snotite. It really looks like barren rock when you're walking across it that's embedded in ice. This is Ice Lake Cave at Craters in the Moon National Monument. But when we incubated slabs of sterile rock on the ice, within 6 months, we got something like this, where you can see some of the individual minerals in the glass that are not colonized by life, but you get these huge microbial communities that have formed. But if I were to show you a slab of that rock, and I will in a minute, you would think that it was barren. And so this really got me thinking that maybe we've been making the whole question too hard. And so I backed up, and right around the time that I was pondering this, Daniel Gezi put out this great study. So he was looking at silicic caves. And what he did was he went and sampled the microbes in barren rock, essentially on the left-hand side, all the way through to a well-developed microbial mat on the right-hand side. And the different colors in this bar graph represent different types of microorganisms. And so what he shows is that the microbial community just changes as these speleothems develop. So between going from the background to these visible microbial mats. but that there are microbes present in all cases. So at that point, I sat down and I went through the literature and wrote down every single study that I came across on lava tube caves and what they sampled. And what I found was that the majority of the work that has been done has been focused on these visible microbe structures and speleothems. And only a handful have focused on that background community, either in snow or ice or rock samples, or in my case, rock samples and incubated basalt chips. And so one area of research that I think we have to pursue in the process of moving towards flying a cave mission is better understanding that background microbial community, because that could be anywhere in the cave. And that would actually make it easier to hunt for life in a very tough set of circumstances, which we'll get to in a minute. But I just kind of wanted to prime the pump that this is where I'm at and what I think we really need to do moving forward. So a little bit about my work at Craters in the Moon National Monument. So I went here because some of the rocks, the basalts on Mars have a ton of phosphate. And in a lot of my past work, nutrient limitation ends up being a critical factor in how much life exists in an environment and what type of life exists in an environment. And so I thought, well, this would be a great place to really look at how phosphate influences microbial colonization, phosphate in the rock. And so we went to Craters of the Moon National Monument, which are these beautiful basaltic eruptions. They're really young. They're only a few thousand years old at the oldest. And they really form as part of this Snake River Plain hotspot track that, you know, sort of burned through from the Columbia River basalts on the very west coast of the United States, all the way through their neighbor to the east in Idaho. And then ultimately the hotspot is currently located at Yellowstone National Park. And so this location has a couple of other things going for it. It tends to be on the cold side, just generally. I used to live in Idaho, and it was one of the coldest places I've ever lived. It also tends to be incredibly dry. So, you know, 200 to 400 millimeters a year, and probably going to get worse with climate change. And then it has this high phosphate basalt composition, similar to what we've seen in some of the Mars rocks that we've analyzed. So it seemed like a good analog environment to kind of look at some of these processes. And so we looked at 4 caves, and we were looking at how does water availability affect things. So from a lot of water, in this case, Pond Cave has ice in the winter and a pond in the summertime that's quite active. And then in the lower temperature regime, we have Ice Lake Cave, which is a frozen sheet of ice that exists across the entire base of the cave. And in the winter, you get groundwater seeping in and actually melting that top surface and interacting with it. And then in the summer, it's completely frozen. It's on an opposite airflow cycle to the surface. And then on the right-hand side, in the low water availability category, you have Screaming Jaws of Death, where water is never observed, though it is present. Like we can see that the soil gets damp, but you can't quantify the water in any way. And then we have Spongy Floor Cave, which intermittently has ice, and then sometimes it's completely dry. And the temperature range here goes from like 17 degrees C to 0 degrees C, or even -1 with the updated data. So we incubated these sterilized basalt chips that are primarily glass with those plagioclase and olivine. And so you can see a scanning electron microscope image of what that looks like on the bottom here. And what's interesting is when we retrieve these after 6 months and a year of incubation, they still look like barren basalt slabs. Like if I handed this to you, you'd be like, it's a rock. Like, why are you giving me this? But when we looked at them with the scanning electron microscope, you actually see these very well-developed biofilms have formed with microbes all over the surfaces of these basalts. So these are both from screaming jaws of death. And the main difference between 6 months at the top and a year is that you increase the amount of microbial activity as quantified by adenosine triphosphate, or ATP, which is that energy powerhouse of this all cells that we have observed. And so this is what that looks like. On the right-hand side, you can see microbial activity increasing with time. Screaming jaws of death has a much different trajectory, probably because it's significantly warmer than the other caves, which sort of group more closely together. But definitely the longer that microbes are hanging around, the more microbial activity you get, the more biofilm coverage you get, things like that. However, the total DNA content, again, screaming jaws of death is doing a little bit of its own thing, but for the most part, kind of levels off and remains the same from that initial incubation through 1 year, and in most samples kind of matches what the background looks like. So what this says is that different biosignatures, DNA is a very high biosignature, ATP would be, but it doesn't hang around very long. It doesn't get preserved in the rock record because it's an active energy transporter. But different biosignatures are essentially behaving differently in caves, and that's further complicated by some of those cave processes like speleothem development. Okay, so the background community is fairly active, and you know, we can quantify biosignatures from it, even if it doesn't look pretty, and even if we wouldn't with our eyes necessarily pick those samples. So how does any of that translate into a cave mission? Well, this is where we get into sort of that opportunities and challenges. And a lot of this work is informed by the work that the MACIE science team did, and I'll list everyone out at the end of my talk. But I do want to make sure that I give them credit up front, because they have really done a ton of work, not just in developing that concept, but actually subsequently. And so a lot of what I'm going to show you comes from a combination of both our work and the work they've done since we wrote that initial concept. So if we want to explore a planetary cave, obviously the first thing we need to do is find a hole in the ground. And it turns out that's actually fairly easy to do. So if you look at this great schematic that Tim Titus put together in Nature Astronomy last year for his roadmap of planetary caves, you'll see that there's a number of planetary bodies, including icy and ocean worlds, that have holes in the ground that we could access and explore. Now, for the moon and Mars, we know quite a lot more about those holes in the ground, just because we've been exploring both those planetary bodies longer, and Mars is the most heavily studied planet, probably beyond Earth. And so we're starting to characterize what those cave entrances look like with the Sherrod data and with high-rise imaging. And we have some amount of handle on what the caves look like, which gets us toward putting together an exploration process, whether that is robotic or human driven. And you can see that both examples are shown in the image. And so this is really where we're hoping to go. But ultimately, the things that we learn about lunar caves, if potentially we fly Laura Kerber's mission concept to a lunar cave moon diver, or if we look and explore a Mars cave, will hopefully eventually benefit some of these other planetary bodies and better inform us of how to look into the subsurface elsewhere. Okay, so where's the best place to look for life? It's going to be three things, and I'll show you why in a second. But warmer caves might be better. We need to do additional work to confirm that because we have a very small sample set. Temperature decreases with traverse distance, and where you sample in the cave matters. So it turns out walls and ceilings are better. And then radiation environment is not as bad as we thought. So we used to think you need to walk a fair distance into a cave to get away from any potential radiation coming from the subsurface. It turns out that within 5 meters of the center of the entrance, you're pretty well shielded. So warmer caves. So back to this ATP, right? So this ATP, ADP interaction where you're stripping or adding a phosphate group really underpins all of our energy metabolism. And that's true for all life as we know it. And so when we measured the ATP content in the caves at craters in the moon, what we found is that as you get warmer, you have more microbial activity. And so the error bars that are horizontal reflect the variation in temperature in the caves, and then the vertical error bars reflect our very small sample size. But still, there's enough here to suggest that this is a trend. And it makes sense, honestly, right? Because you're more active when it's warmer, or at least I am. Okay, so how does that relate to traverse distance? So Twin Falls High School put together this cute little poster that was actually really incredible that I came across at Craters of the Moon one day when I was out there working, because they had a little a poster session, and they had gone through and systematically mapped the temperature variation in Indian Tunnel as a project. And they measured ceilings, walls, and floors. So the three surfaces that you might target, right? And what they found was that, you know, obviously right at the entrance, the floor of the cave is going to be the warmest. And that seems, you know, pretty obvious and intuitive. But what's really amazing about this data set and why I was so happy with it and added it here is that the ceiling and the mid-level walls of the cave remain warm for fairly long distances, out to almost 20 meters, which is, you know, a fairly significant traverse distance for a cave mission, given that it's so hard to land on Mars and to operate on Mars. And so if this holds true for all caves, then what it would mean is that really targeting the ceiling and the walls, you know, so looking at it, maybe scanning it with a laser instrument or something, could provide us a lot of information about biosignatures and life and habitability fairly close to the cave entrance, which ultimately is good for reasons I'll get into that are engineering related. But this is fantastic. And also, like, high schools, man. I am so stoked that they did this. So then, in addition to that, work that was done recently by Faides Moreras in 2021 mapped out the radiation, well, modeled the radiation environment of Mars, looking from that center point of, you have this hole in the ground, and then with lateral distance away from that. And so within that first 5 meters, you actually get to fairly negligible radiation environment that would be clement even for people. So there's quite a lot of shielding. So if you have warmth and you have shielding and you have high microbial activity, you might have a ton of biosignatures that you could sample for a cave mission. So where are the best places to access? This is where it gets complicated, because to have a successful mission, you have to do great science. NASA or ESA or any other space agency is not going to give you $1 billion if you are not doing great science. But there's a constraint on your science that has to do with engineering complexity, which influences cost and risk. And so what type of cave? So we talked about, lava tubes and pit craters, all of those things, but also the shape of the entrance, the breakdown piles, all of those things influence what kind of a robot you can send, and thus what your cost is and your risk. If you have to go with a fairly complex robotic architecture, then you have less budget for science instrumentation, and that can hit your science return. In addition to that, reducing cost and risk is critical for convincing an organization to give you $1 billion to go do this, because the last thing they want is for you to get the spacecraft all the way there and have it fail. So there's only really been significant characterization done of 2 volcanic edifices, Archaeamons and Albamons. But in general, when we were scoping out MACIE, we looked at Earth and we said, okay, what are the types of cave entrances we see on Earth? And so you get vertical entrances, which are near the right-hand side, which are hard to access. And they can either be rocky or smooth, all the way down to the bottom, and can have breakdown piles associated with them. You can also have horizontal entrances, so you could walk in, but like Screaming Jaws of Death, you had that big rubble pile right at the front that you have to navigate around. Or, you could have just a very low slope entrance that you just wander into. All of these things exist. And based on what we think we know about Mars, we know for sure the vertical entrances exist, and that there have been a handful of horizontal entrances that have been observed. So again, we know some of these features are laterally contiguous, but those entrances might be vertical. And then we have this study from Kearney et al. 2021, where they actually went through and characterized the types of entrances associated with Archaeamons and only found 1 lateral entrance. So by and large, the majority of all these black dots are probably vertical entrances, which means we're in that high complexity side of the box. So what does that look like for robots? So, obviously on the left-hand side of the diagram, you have legged or wheeled rover systems that could just drive right into that lateral cave entrance. And that would be best case scenario because it's the least complex and thus the most money available for your science. As you move to the right-hand side, things get more challenging. Maybe the flying robot is not as challenging. But you can see that, you know, in the case of the helicopter, your science returns might be hit because your payload has to be small because you're flying a robot into a cave and there's less atmosphere on Mars than there is on Earth to support that. And so depending on, you know, what cave you select, you may end up with a combination system where you have a lander, a tether, and then your entry vehicle. And at least for MACIE, we were looking at, you might only be able to afford to hang over the side of the cave and look in, which could impact your science. The other thing that we're facing is that the majority of the missions that have flown have landed at low elevation. If we want to land someplace like Tharsis, obviously higher elevation, less atmosphere to slow you down on the way of the surface, that makes it a more challenging landing, and thus it's more expensive and harder to do. And lastly, you know, planetary caves are still classified as special regions. So this figure comes from a study where they were looking at, you know, could we lower the planetary protection requirements for Mars missions in some places in order to reduce cost? And it turns out that they have decided, and I agree with them, that because of the astrobiology potential of caves, you have to adhere to the strictest planetary protection protocols. So that instrument package, that payload and mission needs to be pretty basically sterile upon arrival. And that is very expensive to do and hard. So where do we go from here? The reality is there's a bunch of mission concepts in development that are looking to try to tackle the engineering challenges of how you get into a cave. You know, we may not get this luxurious option of wander in. But also we need to do that additional work on the science to better understand that microbial community. And if we can only hang over the side and look a couple of meters in, you know, can we actually address whether there might be life in the subsurface? And if not, how much would it really cost us to get a payload through one of these vertical entrances and into a cave? And with that, I'd like to thank the wonderful people associated with my Careers in the Moon study, including the folks at the National Park Service and the NASA Astrobiology Institute scholarship, Lewis and Clark scholarship that supported my work at Craters of the Moon. And then all of the people, and I think this is maybe not even an exhaustive list, who helped with the MACIE mission concept, which ultimately underpinned a lot of what we talked about today. And with that, I am happy to take your questions.